Okay, so in this video we are going to discuss about genomic genomic organization of bacteria. So before we start, you need to know what is genome. Genome is simply a genetic material, and we are discussing bacteria. So genetic material of bacteria is called genome. Genetic material. I just forgot word material. Genetic material of bacteria is called this one, and you will see that bacterial genome which is present, it's like if I just make a rough picture. If this is the entire bacteria, you will see nucleus like this. Okay, then you will see one more thing over here. It's like you will see a region near to this one. So this region around DNA, that's called as nucleoid. So this is called as nucleoid. So we were discussing that genome is genetic material of bacteria, and specifically bacterial genome is called as nucleoid, and that's composed of single cells. So this means in nucleoid you will see only single DNA. Okay, and like of course in prokaryote we don't have organelles like cell membrane or nucleus. So you'll find these all material in cytoplasm. Main thing you'll find in cytoplasm, and even in cytoplasm you will see near DNA we'll find a lot of structures like this nucleoid structure which have single stranded DNA. So I'm sure it's clear to you. Then even these all things you cannot see, this nucleoid region you cannot see with your like these naked eyes. You need particular microscope like electron microscope to see. So this means visible through electron microscope. And bacterial, now if we talk about bacterial chromosome, bacterial chromosome is like present in very few number. They're few in numbers, even we just call they have one, but that's long in structure, and they are single molecule of double stranded DNA. They're single molecule of double stranded DNA, and you will see that they are helical and supercoiled DNA, helical like shape, and are supercoiled. Okay, I'm sure it's clear. And no nucleus would be present in this bacterial genome. No nucleus. If no nucleus, so it's like they're openly present in cytoplasm and in their structure. They have circular DNA, as I just mentioned, circular double stranded DNA. I'm sure my writing is not looking very good today. Well, you will see that they are haploid due to single chromosome. They are haploid in their structure, haploid due to single chromosomes. Okay, so it's like helical and haploid. This structure would be like n in number. This would again like divided into these 2n, and in the end they will become like these. Okay, so I'm sure it's clear to you. Then you will see one more structure. As I just made a structure, I'm making a structure again. You will see DNA here. Okay, and here you will see few other things. These are called as plastid. And now if we describe this a little more, so these help in protection, they help in growth, like division, of course not growth like increase in size, but just growth by cell division. And they help in metabolic activities. They have extracellular DNA, extracellular DNA, and they are also called as extra non-chromosome. This means not very important work, but it's still present. Non-chromosome, extra nuclear, and they have non-chromosome DNA like structure. They also help in protection. I mean, they helps drugs resistance, antibiotics resistance, and antibiotic drugs, heavy metals, help in protection, and resistance. This means avoid. Some shots clear to you. Then even yes, one more thing, that their DNA is circular and bacterial nucleoid. This is a very important point. I don't know how I just forgot, because this could be an MCQs too. So I'm just writing over here, bacterial nucleoid. They contain so many genes, even around, it could be around 3 500 genes as well. And in eukaryotic cell you will see that there's no nuclear membrane or nuclei, but in bacteria you'll find one more copy of DNA, as I just told you. So in addition, the bacterial chromosome are like so many. They have DNA. This plasmid is also called as extracircular DNA compound or DNA structure called as plastid, and they usually between 5. Now I'm just talking about this plastid. There could be 5 to 100 genes. Okay, and plasmids help in replicating and provide the conditions bacterial use in, and also used in rDNA technology in carrier of gene. Okay, which is not a topic, it's like a little advancement, and of I'm sure it's clear to you. But before we even complete this, a basic difference between bacterial DNA and eukaryotic DNA. Here is bacterial, here will be eukaryote. So in bacteria you'll see this is circular, here you will see it's linear, and in this structure you will see these are mRNA, and here you will see mRNA but only exons. So exons are like functional part, and you will see introns. Introns are non-functional part which are not important. So this means introns are not present in this bacterial, but here you'll see both are present, and when both will be present, so we call as split gene. Okay, so I'm sure it's clear to you. Thank you for watching. Bye bye.